working in two therapeutic areas. There's new antibiotics to address antimicrobial resistance and oncology. And that, if I understand correctly, it's using sort of the same molecular function. So I'm wondering if you can just talk or similar molecular functions. I'm wondering if you could just talk to us about these programs in these areas, and then maybe we can return to sort of the nature of the problems that you focus here, and like how you think about optimizing for these different therapeutic areas.
Yeah. Yes, that's right. Those are our two therapeutic areas of interest. I mean, in some ways, that they have some similarities. They're both about ultimately, killing or stopping the growth of particular cells, but in one case they're aberrantly proliferating human cells, and in the other case they're undesirably proliferating bacterial cells at some infection site, whatever. So there are some levels of commonalities, but of course the individual approaches are quite different. The targets, even if they're similar, are different in those two organisms, and so you have to deal with that. I guess in terms of the, I guess actually probably a useful one is, if your target, as they often usually are, is inside your protein target is inside the cell, the molecule needs to get inside the cell to reach that target, and so actually that occurrence, getting sufficient concentration of the drug inside the cell for long enough to have the desired effect, is quite a challenge, particularly, I guess, in the bacterial programs, because bacterial cells are very different from human cells. Their structures vary from organism to organism in ways we don't have time to discuss, but essentially they have a complex cell envelope or wall around them that is a very significant barrier for small molecule drugs, which is what we deal with, getting across that barrier into the site of action. And discovering new antibacterials has proven to be extremely difficult. Not many new antibacterials have been approved in recent years, and almost always this lack of accumulation into the cell is what is the failure. It's very rarely failure to find a drug that a compound that doesn't bind strongly to the target. It's overcoming this formidable cell wall barrier that's the main issue there. And there are other similar issues, but to much lesser extent. So that's a clear differentiation in terms of the design approach. With these two projects, we've had to think much more about this issue of cellular penetration in the antibacterial projects than we have in the oncology projects.